Have you ever wondered why healthy cells know exactly when to grow, and just as importantly, when to stop? Every day, billions of cells in our bodies divide to replace worn-out cells, repair damaged tissues, and maintain normal organ function. Yet this remarkable process is far from random. It is controlled by an intricate communication system that tells each cell precisely when to divide and when to remain inactive. One of the defining characteristics of cancer is its ability to ignore these instructions. This ability, known as sustained growth signaling, is one of the original hallmarks of cancer described by Douglas Hanahan and Robert Weinberg. Because this hallmark encompasses several important concepts, we'll explore it over multiple videos. In this first part, we'll focus on the fundamental principles and definitions that lay the foundation for everything that follows. Let's begin with the first image. Unlike cancer cells, normal cells are remarkably disciplined. They do not divide simply because they possess the machinery to do so. Instead, they depend on growth-promoting signals coming from their surrounding environment. These signals act like permission slips, informing a cell that conditions are favorable for growth, tissue repair, or normal maintenance. Without these external instructions, most healthy cells remain in a resting state rather than entering the cell cycle. These growth signals originate from several sources. Many are diffusible growth factors, such as epidermal growth factor, or EGF, and platelet-derived growth factor, known as PDGF. These signaling molecules travel through the extracellular environment until they reach nearby cells. Other signals come from the extracellular matrix, the complex network of proteins surrounding cells. Rather than serving only as structural support, this matrix actively communicates with cells, helping regulate their survival, movement, and proliferation. Even neighboring cells contribute to this conversation through direct cell-to-cell contact using adhesion molecules such as cadherins, allowing tissues to coordinate their growth and maintain their normal architecture. But receiving a signal is only the beginning. For a cell to respond, the signal must first bind to a specific receptor embedded in the cell membrane. Many of these receptors belong to a family called receptor tyrosine kinases. You can think of them as highly specialized antennae, each designed to recognize a particular growth factor. When the appropriate ligand binds to its receptor, the receptor changes shape and becomes activated, triggering a chain reaction inside the cell. This intracellular communication process is known as signal transduction. Rather than traveling directly to the nucleus, the signal is relayed through a series of proteins that amplify and transmit the message. One of the best-known pathways is the RSRFMEKERK signaling cascade shown here. Each protein activates the next, allowing information received at the cell surface to reach the nucleus efficiently and accurately. Eventually, this signal reaches the cell's DNA, where it activates genes responsible for cell cycle progression and cellular proliferation. Only after these genes are switched on does the cell commit to dividing. The consequences of this regulation are profound. When growth signals are present, cells survive, proliferate, and contribute to tissue growth, renewal, and repair. However, when these signals disappear, healthy cells simply stop dividing. They enter a resting state known as quiescence, preserving tissue homeostasis and preventing unnecessary or excessive cell proliferation. This dependence on external growth signals represents one of the body's most important safeguards against uncontrolled growth. Now let's move to the second image. If normal cells require external instructions to divide, an obvious question arises. How do cancer cells escape this dependence? The answer lies in a special group of genes known as proto-oncogenes. These are completely normal genes found in every healthy cell. Their proteins play essential roles in transmitting growth signals and promoting controlled cell division whenever it is needed. In other words, proto-oncogenes are not harmful. They are indispensable for normal growth, development, and tissue repair. Problems arise only when these genes become altered. Once activated by specific genetic changes, a proto-oncogene is converted into an oncogene, a gene that continuously drives cell proliferation even when no external growth signals are present. Unlike tumor suppressor genes, which generally require loss of both copies to lose their function, oncogenes behave in a dominant manner. A mutation affecting only one copy is often sufficient to produce excessive growth-promoting activity. This transformation can occur through several different mechanisms. One possibility is gene amplification, where additional copies of the gene are produced. The protein itself remains normal, but the cell manufactures far too much of it, overwhelming normal regulatory controls. A second mechanism involves point mutations that alter the protein structure, creating an abnormal protein that remains permanently active. A third mechanism results from chromosomal rearrangements, where segments of different chromosomes become fused together, producing hybrid proteins with entirely new or deregulated functions. Scientists have identified several processes capable of producing these changes: random point mutations, chromosomal translocations, viral insertion into the genome, gene amplification, and even epigenetic alterations can all activate proto-oncogenes and convert them into oncogenes. Once activated, the consequences are dramatic. Cells gradually lose their dependence on external growth signals, continue progressing through the cell cycle, survive under stressful conditions, accumulate additional mutations, and ultimately contribute to tumor development and cancer progression. These concepts have also been demonstrated experimentally. When normal cells are grown in culture, they require growth factors in their medium before they will divide. But cells carrying activated oncogenes continue proliferating even when those growth factors are removed. This simple experiment provided compelling evidence that oncogenes allow cells to generate their own growth signals, establishing one of the central principles of modern cancer biology. Now let's move to the third image. The difference between healthy cells and cancer cells becomes even clearer when they are compared side by side. Healthy cells divide only when growth factors are available in their environment. If those signals disappear, they simply exit the cell cycle and enter a resting state known as G0. Cancer cells, however, behave very differently. Once they acquire growth autonomy, they continue dividing regardless of whether growth factors are present, allowing tumors to expand even under unfavorable conditions. To understand how this happens, it helps to examine a typical growth factor signaling pathway. The process begins when a growth factor binds to its receptor on the cell surface. The activated receptor then stimulates intracellular signaling proteins, which relay the message toward the nucleus. There, transcription factors activate genes responsible for producing proteins that drive cell proliferation. Under normal circumstances, every step in this pathway remains tightly regulated and depends on external stimulation. Cancer cells can disrupt this system at multiple levels. Some produce excessive amounts of their own growth factors, creating an autocrine loop in which they continuously stimulate themselves. Others increase the number of growth factor receptors on their surface or acquire receptor mutations that leave these receptors permanently switched on. Still others develop activating mutations in intracellular signaling molecules such as RAS or lose proteins that normally suppress these signaling pathways. Although these alterations occur at different locations, they all produce the same outcome: continuous signaling that promotes persistent cell division. The biological consequences are profound. Normal cells remain dependent on their environment, stop dividing when signals disappear, and remain under the control of surrounding tissues. Cancer cells become independent of these external controls. They continue growing, survive in hostile environments, and acquire an increasing likelihood of accumulating additional genetic alterations that further accelerate tumor progression. In this first part, we've established the fundamental concepts behind sustained growth signaling. We've seen how healthy cells carefully depend on external growth signals, how proto-oncogenes normally regulate this process, and how their conversion into oncogenes allows cancer cells to escape one of the body's most important growth control mechanisms. In the next part, we'll build on this foundation by examining the major oncogenes involved in human cancer, including EGFR, HER2, RAS, MYC, and others, and explore how their discovery has transformed both our understanding of cancer biology and the development of modern targeted therapies.